and the interconnected pathways between the different regions, all these white matter tracts that connect the different brain regions. So at this level, already the structure of the brain is obviously very complicated because we know all these different regions of the brain are highly interconnected. But if you zoom in into one of these particular regions, like say motor cortex, you will see a very intricate connectivity. This is not really how it looks. There's not so much space. But you see these 6 layers of different neurons, different cell types, and we know a lot about the interlaminar connectivity, cell type specific connectivity. So the complexity at this mesoscale is also well known and established, and we know a lot about it. So we're seeing already 2 different levels where there is a lot of complexity. But then if you go in furthermore and you zoom in into one individual neuron, you again see this massively complex machinery where you have all these ionic channels, you have synapses, different molecules interacting. And so what we think, or one of the things that makes the brain so complicated, is the interaction across all these scales. So of course, what is happening at this molecular level, cellular level, is affecting the dynamics, the responses at the neuron, at the circuit level, and even at the macro scale whole brain activity, right, with oscillations, for example. Let me enter full screen. And this happens both ways. So the molecular level affects the cellular circuit and whole brain, but also the whole brain dynamics affect the cells and even the molecular changes and so on. So this is very complicated, but we are lucky, I feel, because we live in an era with an unprecedented amount of experimental data. And this has been coming out of many different projects. In the US, for example, you have the Brain Initiative, which is getting data at a level of resolution that we didn't have before. I'm showing this electron microscopy. This is from the mouse cortex, and it's 1 millimeter cubed reconstruction of all the neurons and all the synapses that you could find in 1 millimeter cubed of the mouse cortex, around 200,000 cells, 500 million synapses. And you can see the exquisite level of resolution that you have for this dataset at the synaptic level. And interestingly, you also have corresponding physiological data, so recordings from these same neurons in response to different stimulation, for example, watching like natural videos and so on. So you really have both the anatomy and the physiology with an incredible level of resolution. Fascinatingly, there's also a similar dataset for human cortex that was recently released as well, with 50,000 neurons, 130 million synapses, and here you see a nice visualization of the different neurons across the 6 layers of cortex. And this is from that same dataset. This is one single human cortical neuron with, I think it's like 5,000 axons that were projecting to this neuron. Again, with a fantastic level of detail. Of course, we also have a massive amount of physiological data. So here I'm showing like also the different scales at which you record this data. So you have from the more abstract macro scale EEG down to ECOG, LFPs, extracellular recordings, and intracellular measures. And at the same time, we are gathering simultaneously behavioral data. So while you record all these signals, for example, LFPs, you can record the behavior of the animal to try to figure out the correlates between behavior and neural activity. So how can we make sense of all this crazy amount of data from different modalities, anatomy, physiology, behavior, recording from different scales? Well, we think one approach is this idea of multi-scale circuit computational models. So these models basically allow you to integrate all these different data at different scales. So you can, incorporate morphologies that you record, that you measure. You can reproduce the activity of neurons at a very detailed level. You can incorporate pharmacology, biophysics. You can even reproduce some behavioral paradigms. And so these tools really, if you are able to construct a model with enough constraints from experimental data, and that validates or reproduces the activity that you see in the real brain, then you really have a small virtual brain in your computer that you can then poke and play around to try to understand the mechanisms behind the activity that you're seeing. So that's sort of the pitch of what we do. And now I'm going to show 3 different models of this type. I'm going to focus and spend a bit more time on the first one. And the other ones are, similar approach, but applied to other regions. So this is the motor cortex model. So this model represents a kind of cortical column in motor cortex. So it has all the 10,000 neurons that you would find within this volume of tissue, and around the 30 million synaptic connections you would find there. We work with an experimental lab that provided us all the data, Gordon Shepherd's lab. So they have data about morphologies, about the physiology, connectivity, lots of very detailed connectivity data. And we put everything into a single computational model. So to give you an idea, this is how we build the models for each single neuron or cell type. So here you see the experimental data for the morphology and the reconstruction, the simulation in the computer. As you can see, it's practically identical. So you have all these different compartments that you can simulate the voltages for. Here we have the experimental data for these FI curves, so current injections at different amplitudes and the responses that you measure from the cells and how we reproduce this in the simulations. To achieve this, basically, we are playing around with the conductances of the different ionic channels. So this model has around 10 different channels, sodium, potassium, HCN, calcium channels, and so on. And we play with the values within known biological constraints for each of the channels and distributions along the dendrites, and use supercomputers to optimize these models to the data that we're seeing. So they are basically feasible models of the experimental data that we see. And we match both these FI curves, but also subthreshold responses and so on. And we do this for all the cell types that we find in this circuit. So in this particular circuit of the motor cortex, we included 3 main excitatory cell types, these IT or intratelencephalic cells, which can be of 2 classes, corticocortical and corticostriatal. And those are present in all the layers, layers 2-3 to layer 6 are these red ones. Then you have the PT or pyramidal tract projecting, which are mainly these corticospinal neurons, which are only in layer 5B, and they project axons to brainstem and spinal cord. They're kind of the output of the system here. And the third type is these corticothalamic neurons, only in layer 6 mostly, and they project to thalamus. Then we included 2 types of interneurons, although in more recent versions we have 2 additional types. So here we have this somatostatin and parvalbumin, but we also have the neurogliaform and VIP cells in neural models. And we distributed these cells based on known experimental data. So we have the density of the different, or the density of neurons in each of the layers, and you can see that here. We have the proportion of cell types that you find in each layer, and so we incorporate all this information into the circuit model. In this case, just in case you're wondering, we didn't model the morphologies in detail for all the cell types, only for 2 cell types that were of more interest, this layer 5A corticostriatal and the corticospinal ones, and the rest were modeled using these simplified morphologies. In this case, it was for computational costs. Running these models is really heavy. So these simplified morphologies still capture a lot of the interesting phenomena, but allow you to run more simulations. And then the other interesting part where we included a lot of data is the connectivity. And we have connectivity at multiple scales, as I was showing before. So on the one hand, we have all the long-range inputs that come into motor cortex from other regions. This includes cortical regions, like somatosensory cortex, secondary motor cortex, and so on. And also 2 different thalamic regions, the motor thalamus and the sensory thalamus, like they call it, VL and PO. And each of those, we have really nice data showing they target specific layers of M1. and specific cell types. So, if they go to layer 5B, they might project only to the PT cells and not the IT cells, for example. So we include all that data as well in the model. Then we have a lot of very nice data for the local microcircuit connectivity. So how the different cell types within the circuit connect to each other, based both on the layer of the cell and the cell type itself. And that's also very well known. We even found differences within a single layer, differences in connectivity at the top and the bottom of the layer. So for example, layer 5B, there's stronger connectivity to some cells when they're at the top versus the bottom. So this is like sublaminar level connectivity patterns that we also incorporated. And finally, we included the dendritic distribution of these synaptic inputs for each projection. So for example, for layer 2-3 IT cells to layer 5B PT cells, the synapses are mostly in, around the soma and then some in the apical dendrite. So this is almost like a probability distribution of the synapses. And we didn't have this for every projection, but we included the ones that we had. Okay, so we have this connectivity at these 3 different scales, and then we submitted the model for publication, and they said, okay, very nice, you have a lot of data, but we need more, because we need to know that the model is actually doing something or reproducing some real stuff. So then we had to go back and try to validate the model against in vivo data. So the reviewer said, you need more validation. And so we contacted these experimentalists in Edinburgh, Ian Duguid, who did really nice experiments in mouse M1. And he did in vivo experiments, recording from different cell types intracellularly, which was exactly the type of data that we needed. So we could know, the voltages, but also the firing rates of different cell types in M1 and their different behavioral conditions. So this, allowed us to link our model of the circuit to also behavior, which was super interesting to us. And so the behavior that we were modeling is this free walking on a treadmill. And so based on the literature and so on, what we saw is that the changes that happen in the brain, the main changes that happen in the brain when the mouse is running versus quiet or not moving, are 2 inputs from motor thalamus to M1, lateral thalamus, they increased, and inputs from locus ceruleus to M1. So we modeled that we already had that connection, so we just modeled 2 behavioral states. One is the quiet state or spontaneous, and one is the movement state, where we basically increase these 2 long-range inputs to M1. Each of those go to very specific cell types and have very specific phenomena, like the locus ceruleus changes the HCN conductance, for example. So we incorporated these things. All right, so now we have the circuit, we have some link to behavior, and we were able to start validating the model. So here is a raster plot of the 10,000 neurons in the M1 model. Different colors represent different cell types. The red ones are the ITs in different layers, blues are the PTs, and the greens very light here are the CTs, which don't actually fire much in real life either. And we were able to simulate these 2 behavioral states, the quiet state and then the movement with these increased long-range inputs. And so we validated the firing rates for these 2 different states in the experiment and the model. And you see here there's some match statistically, showing the changes that you see in different populations. They match between the experiment and the model. So this was kind of our first validation of the model. And the second was looking at local field potentials, LFPs. Similar setup, these are LFPs recorded from layer 5 in the 2 behavioral states, and we compared the model to the experiment. And here, of course, we don't expect to see exactly the same pattern, but mostly statistical properties of the signal. And so we saw that during the quiet state, we have mostly slower oscillations in the delta range frequency, and during movement that increased to gamma. And so we validated that statistically as well with multiple simulations where we simulate also different trials in the model. So we have like some statistical power and different trials, different animals. We can do some variation in the model to simulate this. And we showed some additional validation of the LFP signals. Then we also looked at this stuff because one of the reviewers asked for it. So we didn't have any clue about what this stuff was, but it seemed interesting and they wanted to see it. So this is what they call low-dimensional neural manifolds or representations, and it's been shown to correlate to behavior. So it's basically you take the 10,000 neurons and you collapse it into 3 dimensions. So it's a low-dimensional representation. You can use different algorithms. And that seems to correlate very well with the behavior of the animal in many different species and many conditions. So, mice going in around a maze, you can predict where they're going to go based on these representations. Monkeys doing a reaching task, you can also predict exactly the direction and so on. So it seems that maybe the brain is employing some of this, dimensionality reduction to relate to behavior. We don't know, and so we basically apply this to our model, and we collapse the activity of the neurons into these 3 dimensions and for the 2 different behavioral states. And we saw some nice results. So you can clearly differentiate these 2 clusters of activity. You have the quiet state here on one side and the movement on the other. The movement even showed these cyclical patterns which correspond to gamma oscillations. And so it was a nice also validation. And we're actually trying to validate this directly to experimental data now. So we can calculate these low-dimensional manifolds also in the experimental data and try to show that the model corresponds to what we're seeing experimentally. So this is nice from my perspective because it's the most related measure to behavior that we have. So it's kind of a proxy for behavior in a way. And so it's getting the circuit model a bit closer to behavior. All right. So we have our model, we have validated it partly, and now what can we do? Why is the model useful? So of course, we want models to make predictions. Otherwise, they don't have any value. And so one of the things we did is try to predict the sources of the LFP oscillations that we were seeing. What are the different cell types that are generating or involved in these oscillations? So the one that we saw for the quiet behavior is a sort of delta oscillation that we see here. And with the model, of course, you have access to everything. So you can know exactly what generated that LFP's signal. And so we calculated which are the cell types or populations that contributed most to this LFP oscillation. And these were the 6 populations, so layer 6 IT cells, layer 5 B IT cells, and so on. And we can calculate the LFP signal generated by each of those populations isolated, which obviously is something that is not possible experimentally. And what we saw was quite anti-intuitive. We didn't expect to see this, but we saw the 2 top populations were almost in antiphase. So you see here, this is layer 6 IT and layer 5 B IT, and they are not quite, but almost in antiphase. Which is interesting because then when you look at the combined signal, of course, this LFP signal comes from the combination of all these smaller components. And the noise that you see in the overall signal and the fact that it's not a clean oscillation might be a consequence of the signals that contribute to it not being aligned, not being in phase. So that's kind of a prediction of the model. And then the other thing we predicted or tried to predict was what's going on when you do some manipulations in this setup. So experimentally, they had an interesting manipulation where they reduced the input from locus coeruleus. So they reduced this noradrenergic input. And this has been associated with some symptoms of Parkinson's. And you saw some behavioral changes. So the forepaw or the paws were misplaced. And so we tried to come up with an understanding, a mechanistic understanding of this through the model. And so it turns out that when you reduce these locus coeruleus inputs, as I was saying before, this affects the HCN channel, so conductance, they increase, and these are mostly in the apical tuft. So this in turn reduces the synaptic integration of inputs coming through the apical tuft, and this results in a reduced firing of the corticospinal cell. So these neurons are the ones that project to spinal cord. So if you have this reduced firing, it makes sense that this behavioral effect could be produced by this cascade of effects. And so this was a nice link of the different scales from the molecular level, cellular level, to the circuit level, and up to behavior, trying to relate all these different scales. All right, so now I just want to mention quickly a few other applications that we're using this motor cortex model for. I'm not going to go into the details, but just to illustrate some of the things you can do with them. So we have a grant looking at decoding of brain-machine interfaces from motor cortex. And so the cool thing about these models is that you can generate measures like EEG as well. So there are very accurate models of these volume conduction models that you have in the brain and in the skull. And you can generate quite accurate EEG signals based on the dipoles of the neurons. And at the same time, we could calculate these neural manifolds that I was saying before that are associated to behavior. So we asked the question, can you decode any information about these neural manifolds from the EEG signals? And if so, maybe those can be used to decode movement and improve brain-machine interfaces, basically. So we're working on this with the model and also comparing to some experimental data. Another project we have is looking at channelopathies. And so this is different genetic mutations that affect, for example, the sodium channels, NaV1.2. And we're working with UC Davis, who had data on these channels experimentally and some simple models. And they wanted to know what would be the effect when you put these mutated channels in a circuit. And so to try to understand that, we have the baseline model, we put the mutation, and we see how it affects the circuit dynamics and make some predictions, for example, related to epilepsy, which is gain of function of these channels. And also then how would be potentially correct for those changes. So we are also trying some pharmacological manipulations and testing drugs in silico that could restore the original healthy dynamics of the circuit. All right. So now I'm going to switch to a different model. This is a model of the somatosensory, cortical, and thalamic circuits. So the approach to build it is similar to what I said before, but this is actually a much more detailed model. This was originally built by the Blue Brain project in Europe, which is like a massive $1 billion project. And it has like 30,000 neurons, 40 million synapses, 200 different cell types, which is crazy. And so what we did is convert this model to our own modeling tool, NetPine. Until then, it had been sort of very closed and not very shared by them, very difficult to use. So we converted to our tool, which is a bit more friendly. And we also extended it to include thalamus. So they didn't really have the thalamic region. So here you're seeing the 40,000 neurons approximately. This top part here is cortex, and the bottom here is different thalamic regions. So you have like the thalamic relay nuclei and 3 different regions, VPL, VPM, and PO. And they are, of course, connected bidirectionally, right? So you have the thalamocortical and corticothalamic connections. And so, you can put this model into different regimes, like more in vivo or more in vitro, awake or sleep, depending on different parameters. And you can generate local field potentials because it has all the detailed morphology, so you get very accurate local field potentials. And so again, we were validating this model, showing the types of oscillations that you see, and LFP patterns match experimental data. And we are now using this model for other projects. So we're first using it to study epilepsy, and in particular, this fast ripple oscillation. So this is a very fast oscillation, like 400 hertz, that you see previous to epileptic seizures. So it's like an epileptogenic marker. And we reproduced these fast ripples in the model, and we tried to understand where they come from. So what particular cell types, populations are triggering these fast ripples, maybe to understand a bit more how the epilepsy is generated. And so we first reproduced, here's the number of fast ripples per minute in humans and in our model. And then we introduced modifications to increase this number. And we also look at the actual activity of the network when this happened. And we try to figure out the mechanisms underlying these fast ripples. With this S1 model, we are also working with collaborators in Toronto, which have a really nice experimental setup where they are looking at transcranial alternating current stimulation for stroke. And they're trying to systematically try to understand what happens when you apply TACS in S1 to rats that, they have recordings from these rats. And basically they're applying like different amplitudes, different frequencies, and really trying to characterize the response of the TACS. So we use this model to validate against this experimental data. This is some preliminary data between the experiment and simulation, and trying to show that the parent rates of different cell types in the model correspond to the measured activity. And again, trying to understand and make predictions about the effects of TACS on S1, and potentially to use it as a treatment. If we can predict what amplitudes will work better for stroke and so on. And finally, for this S1 model, we're also working with Harvard Medical School, where they're using TMS, transcranial magnetic stimulation, for Alzheimer's treatment. And they have really nice phase 3 trials on humans, showing like TMS is working better than any pharmacological approach right now, and reducing the cognitive decline and so on. And they contacted us. They wanted to understand, what is going on when we apply this TMS, because really it's a bit of a black box at the circuit level. So which cells are being modified, what are the dynamics at the circuit at different layers and so on. So we're trying to build like a very detailed, accurate model of TMS stimulation and the effect that they have on the different cell types. So for example, for this, we had to include the axons of the neurons, which we sometimes remove, and we simplify their implementation. But here it's very important because a lot of the TMS effects go directly to the axon, and they have this action potential there. And here's an example of the S1 model with the baseline condition, and when you apply TMS, you see this synchrony of activity happening. And now we're starting to validate against the EEG measures that they have from clinical patients. All right, so that was a detailed model of S1. The thalamus model that I connected there was a very simple one with just what we called single compartment neurons. But one of my students has been developing a very, very detailed model of thalamic circuits in S1. And so he has, this VPM and TRN, different populations with morphologies also for the different thalamic neurons. He has the feedback from layer 6, so this cortical thalamic feedback, and the inputs from brainstem. And he has been able to show different responses to whisker deflection. So because we have the brainstem, we have modeled all these tonotopic inputs and show that we can reproduce. So this is experimental data in the thalamus and in brainstem. And so you show similar response patterns in the model, and also this angular tuning. So we have very clear angular tuning in these thalamic regions. And so we can reproduce these for different angles based on the brainstem inputs. So it's topographically organized, not tonotopics. And also another interesting stuff, interesting result from this model is reproducing the wake and sleep dynamics, which are very prominent in thalamus. So you have to change, some parameters of the model to reproduce the oscillations that you see during the wake state and during the sleep-like conditions. We have like two different sleep states, deeper and less deep. And so the angular tuning only happens during the wake condition, because it's when really the thalamus is able to process information from the brainstem, but during the wake it doesn't really respond as much. So we're hoping to connect this very detailed thalamic model to the very detailed somatosensory cortex model to make a bit more realistic model. All right, so I'll let you breathe a bit, and we're gonna go to the final model. So this is a model of the auditory thalamocortical system, but in macaques. And so this is a schematic of the diagram of the model that we built. Of course, for macaques, there is a lot of missing data. So for rodents, it's easier because there's more data for the different components. For macaques, it's very hard to find. So we incorporated as much as we could from literature, but a lot comes from rodent because the data is just not there. So what we model is primary auditory cortex and the thalamic regions, the medial geniculate body, and the thalamic reticular nuclei. And then we also included a model, what we call a phenomenological model of the lower component of the auditory pathway, going from the cochlea to the inferior colliculi. So what this does is that we can provide any arbitrary sound waves that we use for the real macaque experiments. We can provide it to this model, and it really processes the input signals as the cochlea and these different early stages does, such that the output is really spike times that you would get from inferior colliculus. So it really provides a realistic input of any arbitrary sound. So we can provide that both to the real monkeys and to our model. And here is just showing this idea of how these models can really simulate all the measures across different scales. So this is the model of A1 with 10,000 neurons or so, and we have the membrane voltages for each of the cell types. We have the spiking activity of the 12,000 neurons, LFPs and current source densities, and as well as EEG. So in this case, we have a lot of EEG and LFP data from the macaques. So this really allows us to validate and constrain the model more and more across different scales based on the experimental data that we have. And we are this is some examples of the validation. This is an example of the LFP and current source density in the experimental model, and different oscillation patterns that you see in the model and experiment as well. So, we're constraining more and more statistically as well. And again, what we can do with this is try to figure out the contributions like we did before of different cell types, different layers to some of the oscillation patterns that we see. So here we're just quantifying how much each of these cell types contributes to some oscillatory patterns that we see in the current source density. So making predictions about this that can then be tested experimentally. And we're also using the same model to study schizophrenia. So there are very clear schizophrenia auditory biomarkers. For example, these, in healthy controls, you have this 40 Hz response when you provide a click train, and that disappears for schizophrenia patients. So we reproduce this in the model, taking into account changes that are known to happen in schizophrenia circuits in auditory cortex. And you can evaluate different hypotheses of what is generating these changes using the model. And then we're trying to understand exactly the mechanisms of why it disappears and so on. So, the greater picture of this schizophrenia project is to try to incorporate as much data as we can from both genetics, from PET scans. So there's a lot of data that you can really use to constrain the models, and try to recapitulate different biomarkers within the same model. So before I was showing this 40 hertz auditory steady state response biomarker, but there are like at least 3 others, this mismatch negativity, the P300 and others. So if we try to build a model that recapitulates all of those, then there is more chances that the model is more correct. Of course, all models are wrong, but hopefully some are a bit useful. So we try to make unified models that can capture as much as the experimental data as possible. And finally, as with all projects, we try to explore novel treatments and evaluating different potential pharmacological treatments. And because these models are so detailed, you can really evaluate these things at the molecular level. All right, we got to the final section, which is I just want to quickly show the tool that we use to develop all these models that I have just shown, which is also a tool that we develop in the lab. So the tool is called NetPine, stands for Networks Using Python and Neuron. So Neuron is a widely used simulator in the field, has thousands of applications, and has been going on for 40 years. But it is hard to use when you want to build complex circuit models. So this NetPine tool is basically a software to make it easy to build these complex circuit models, run the simulations, do analysis, optimize the parameters and explore parameters. So basically the whole modeling workflow that you would need to do for this kind of modeling has been funded by the NIH during the last 5 years and hopefully for the next 5. And so the motivation for this tool was basically we wanted to build these models ourselves, and it was really hard. So we started doing this our own little tool for ourselves to incorporate data at different scales, like I was showing before, connectivity, biophysics, morphologies. And then it started to grow and grow, and it's now being used by many labs. So to illustrate, this is Neuron, and this is the kind of code that you have to write to create one synapse. So our tool builds on top of this and provides a very easy language to code and provide the definition of your model in a human readable, simple language where you don't really need to learn to code. And on top of that, we have a graphical interface where you can actually build all the models as well, just clicking around and playing around. One interesting thing, which I think is quite relevant to this group, is the ability to also model the molecular scale. And this is through a tool called reaction diffusion that Robert McDougall develops. We work very closely with them. I know he was here some time ago. And so this allows you to simulate intracellular and extracellular diffusion of ions, calcium, potassium. And you can also define internal structures, organelles inside the cell, like endoplasmic reticulum and so on, and simulate molecular pathways, all the molecular processes, phosphorylation, buffering, second messenger cascades. And then, of course, evaluate the interaction between these molecular level processes and the neurophysiology of the neuron. So how are these changes affecting the cell firing, the plasticity of the cells, and so on, and at the end, the circuit level response. Right? Of course, the tool also allows you to build models very easily, to provide connectivity using different rules and things that are known to happen experimentally, or at least that you record from experimentally, like probabilities of connection, convergence, divergence, these kind of things makes it very easy to build all these models. It provides all this built-in simulation analysis for typical measures that you use in neuroscience, lots of connectivity plots or visualizations. You can record any current, voltage traces, statistics, firing rates, LFPs. Here we see, measuring the LFP for a single cell and for a network. You have built in this current source density analysis as well. And lately, we have added this idea, this ability to record also EEG signals. Like I said before, calculating the current dipoles, which is really what generates the EEG signal on the scalp, current dipoles of each of the neurons in the circuit, and then using these forward conduction models to generate a realistic signal of the EEG that you can compare to experimental data. And for example, if you have EEG data from patients, you can try to figure out what is the biophysical source of that EEG disturbance. And well, briefly, just to mention, we have also a collaboration with another tool for modeling, but this is for modeling at the whole brain. This is called the virtual brain. It's a massive project in Europe, lots of funding, and they model activity at the whole brain scale, macro scale. So they divide the brain into 50 nodes or so and simulate activity in each of the nodes, but in a much more simplistic way. And what we did is connect both tools so that they both run the simulations in parallel and exchange information. So basically, our very detailed circuit model becomes one of the nodes in the macro scale whole brain model and exchanges information. So the circuit model sends information to the rest of the network, and the whole brain network sends information back at every time step of simulation. And so it's really interesting for us because you can see sort of the effects of the whole brain activity in your circuit model, and also how the circuit affects the whole brain. Here's just to illustrate the graphical interface that I was mentioning. So you can build all the models just clicking around and typing in these boxes. Here's showing the different ionic channels of some cell type, but you can build the populations, connectivity, everything. You can run the simulations and visualize your cells and the voltage traces and activity and so on. And here's like a combined visualization with many plots. You can even see things like the calcium concentration in the extracellular space while you look at LFPs and correlate these molecular level changes to LFP signals and so on. And we're very happy that it's sort of taken in the community, and there are many labs now using the tool. Lots of models being built for different parts of the brain, different cortical regions, thalamus, lots of different disease, different treatments, TMS, electrical stimulation, ketamine, these kind of things. And most of these models are open source, so you can just take them, modify them, play around with them, and publish it. So this, again, has resulted in lots of publications using the tool. So we know, I think it's 39 papers now that have been published using the NetPine tool. And it's also being used in a lot of grants. So there's like at least 12 NIH grants that use NetPine for their research. And so if you're interested, we have the very nice website, netpine.org, where you have lots of resources, lots of documentation, tutorials. We have lots of videos, like 40 hours of video tutorials where you can follow along to learn how to use the tool, and example models, and a forum. And we're always happy to interact with you guys to help you get started. We also do a lot of courses and workshops over the year. So like 2 or 3 each year, many in South American countries, but also here. So you can check in the website if there's any available. And that's it. So just to acknowledge all the funding and of course, all the people that did the work. Many people in my lab and collaborators. And thank you so much.
Hey, thank you very much. Oh, questions? Yeah. Okay. That's tremendous. So many important tools that, I can see neuroscience will be, moving forward for many, many decades, further, understanding of brain circuits. I had 2 questions, which is to ask whether the tools you've developed can help us understand sort of the differential contributions of the extracellular dynamics of neurotransmitters. Very, sort of seemingly simple thing, but we know neurons go through bursting states and have changes in tonic activity. And so you mentioned extracellular ions and whatnot, and neurotransmitters weren't on that list, but presumably by modeling some of the patterns you would expect from different release dynamics, you could also give insights into, for example, we work on a mutation that causes a drift upward of extracellular dopamine that basically would mimic chronic tonic dopamine signaling. And the questions about how that affects a circuit would be seemingly very amenable to modeling. And I wondered if the tools you have do that.
Yeah, so the best example we have for extracellular simulations is for ischemic stroke, where we simulate this spreading depolarization of potassium. I think I actually added it here just in case someone asked. Let's see. No, I don't have it. But yeah, you see very nicely the spillover of potassium and how that spreads over time, and it affects more and more parts of the circuit. So it really, it's the concentration of potassium in the extracellular space, how it changes over time and affects the dynamics of neurons in space. So I think the dopamine would be
right. So because you mentioned also some of these intracellular dynamics, which of course receptor activation is going to trigger, and then you're going to have long-term consequences for that. And I wondered how deep that modeling has gone, because ultimately, you get to gene expression and you get to plasticity and changes. So at the level of, okay, I want to tweak an ion channel and change the firing of a neuron and see it propagate, that's very simple. But your tools are really powerful enough to look at larger timescale things as well.
Yeah, there's hasn't been that much work. So the framework is there, and we have examples where you show second messenger cascades or these glutamatergic metabotropic synapses and how they affect like the concentration of calcium, and that activates the endoplasmic reticulum, and there's a release of calcium, and this multi-step process. But I think it's really ripe for applying this to known experimental results and trying to explain them. The potential is there, and it's definitely feasible. So we're trying to find more people interested in this kind of very multi-scale modeling.
Thank you.
I guess that's a kind of an extended question from Randy. I'm really amazed by all of this, and I have a hard time to comprehend really that's what's going on at the simulation level. I get the, for example, you can make a computation of a neuron and change a ion channel on the membrane, or you compute a synaptic network and you change the number of synapses or which neuron activate which. And here you really have this 3D like circuit with all these components that, are taken into account. And so I'm, especially for the LFPs, I'm amazed. So you can like basically simulate an electrode within that column and basically decide where you record. And if so, does that mean that really like your computational models do have all the ion, as you mentioned, like into the model? Yeah. And that goes to such level of complexity that I'm really amazed.
Of course, there are a million limitations because as I was talking to someone before, we don't really know the distribution of these channels in neurons, in cortical neurons. It's really an unknown. So we have estimated them based on FI curves and whatever. But yeah, the LFP measures, they're really, like you say, they're computed by summing the contribution of all the transmembrane currents of different ionic channels along all of the sections of the dendrite for all of the neurons in the circuit. So it's a pretty big computation, but that's what makes it, and scaled by the distance to the electrode, and so on.
Because at the beginning you were showing that you could like control the density of these neurons in the column, so you change those parameters and you change obviously the LFPs, I'm guessing.
Yeah, so for realistic LFPs, you really need the realistic density of neurons, and that's something we see, and the morphology. So in simplified neuron models, you get less realistic LFPs, of course, because you don't have all the dendrite contributions. So, yeah.
Really, really interesting. So first of all, I like how now we can solve schizophrenia and never get out of our pajamas. So that's the good part about this. But I have a question. I like your second slide. When you validated the contribution of all the ion channels to the electrophysiological signal that you have, it seems to me that you could have all multiple ways in this, the way this works. You have like high and low energy conformations or whatever, so to allow it to get to that kind of a resolution. Do you understand my question?
Yeah, I think you're talking about this idea of parameter degeneracy, where you can have many solutions for
Correct.
the same data, right?
Correct.
And this is a question we always get by reviewers and in talks. And that's one of the big limitations of the models. You know, there's no single solution. And so for that particular experimental data, there are 1,000 models that can reproduce it. So which is the correct model? We don't know.
Does it matter in the long run?
That's the point. So what seems to happen in biology is a similar thing. So there is also parameter degeneracy in biology. And you see this with experiments from Eve Marder, where for the same circuit function of oscillations in the somatogastroganglion cells, you have different animals producing very different solutions. So I think the order, the conductances of some channels in these pieces were up to 3 orders of magnitude different in different animals, which is kind of what we can see in the model. They all work, they all produce the same output, but you have this variability intrinsic. So it might be also more of a feature than a bug, but of course we don't also know if the variability that you see in the models is the same as the one in the biology, but it is there.
By the way,
and the other things, just to say, what we try to do is constrain the models by more and more experimental measures. So we not only reproduce the spiking activity, but the spiking plus the LFP, but maybe the EEG and some behavior, so that to try to narrow down the parameter space of the models, so we get closer, hopefully, to the real thing.
All right, thanks.
More questions? Okay, you're closer.
Quick question, I guess. You show that the reviewer were asking you to validate your model based on experimental data. Is there any? I guess that's related to the question we just had. Is there a way to use this simulation to make new discoveries of something we don't even know how it works in the brain, right? When actually a model doesn't match the experimental data, and the reason is maybe because we haven't been able to put new information in that model because we are simply not aware of it.
Yes, I would like to say yes. I mean, most of the way we use the models is to make predictions, and in a way, some of these predictions are things that haven't been seen. Some of them, there is similar stuff already in the literature, but some is like new stuff that we can go then into the experiments, try to use the same protocol or paradigm we use in the model and see if it's there. If it's there, great, because it's a prediction that the model successfully predicted. If not, we have to figure out why not, and that's also interesting. So.
Yeah, I'll ask.
So I'm very interested in learning your view on manifolds. Is it useful, or because this is a discussion that we have been having in our lab, and okay, so you can have those different geometries, they are related to behavior and so, but and also I got the way you presented. So a reviewer asked you to do that. So what do you think about this new trending?
I'm quite excited about it, to be honest. It's been a field that I always saw in the distance. And, I saw all these nature papers that they get. I saw, oh, good job. But I didn't really understand it. And now so what we're trying to do is really link these two types of models, no? The very detailed biophysical mechanistic to these more abstract ones. And it's nice that we can go from one to the other and try to understand where are these neural manifolds coming from in terms of cell types, for example, in terms of layers, which are the main cell types involved in determining this manifold. And that is obviously very hard to do experimentally, and something that also, many experimentalists don't really like that much the neural manifolds because they don't understand them. Like, what does this mean? I have my neurons, and then I have this very abstract representation. So I think we can do like a link between those two worlds with these models and try to have the whole spectrum connected. So I'm excited.
Questions from the chat now. So I'm going to read this to you. It seems like there is a cost benefit in terms of computational data that is available for simulations where the highest resolution data we have for species that have very dissimilar small brains, such as the fruit fly, and the computational data gets lower resolution as we move towards brains that are closer to humans. How do you go about navigating this and choosing what size/species brain you want to look at to study a particular problem?
I mean, it is completely true. So for larger brains closer to the human one, we have less data. So I mean, how to decide? Well, that depends on your collaborators or what is the question of interest or the grants where they come from. I prefer to work where there is more data, obviously, because it's easier for the models and you don't have to make up so much stuff. But it's true that it's also very interesting to work closer to humans because you can make more clinically relevant predictions. So yeah, it's exciting because we're getting also a lot of data for humans, and we have a lot of data for human cell types now in Cortex for connectivity. So I'm quite excited about trying to build models with all this new data, but there's a lot of missing, of course.
Another question, why did you choose M1 to model? Was it based on the availability of data? Do you have any future work in mind? What did you consider?
Yeah. We chose it because our collaborator was the expert in this model, and also because it was not so much overdone in modeling. So in modeling, you see a lot of models for visual cortex for many years, and hippocampus also, somatosensory cortex, but there wasn't anything on motor cortex. And there is actually motor cortex now has become the region with more experimental data available, with a recent publication from the Brain Initiative Cell Census Network, BICCN. They released these 10 Nature papers with super comprehensive data from transcriptomics, patch-seq, everything together, connectivity for mouse M1 and also some macaque and human, but mostly mouse M1. And really categorizing all the different cell types, all the connectivity in massive detail. So I'm really excited about that data set and trying to model that now. Yeah, it's the best known region that we have right now.
Thank you.
Thank you so much.